let us discuss activation energy. In the field of enzyme kinetics, activation energy is the amount of energy required to convert 1 mole of a reacting substance from the ground state to the transition state. What do I mean by transition state? To understand that, look at this. So you have a reaction coordinate and we are going to depict free energy, that is Gibbs free energy in the y-axis. This is your substrate, this is your product. As you can see, the free energy of product is lower than the substrate. So what kind of reaction is this? During the reaction, energy is released. That is why the product is having lesser energy, right? So this is an exergonic reaction, right? For a substrate to become a product, first it has to become the transition state. Transition state is always depicted by the symbol double dagger. So transition state is a fleeting, momentary chemical species. It is neither substrate nor a product. Transition state has the highest free energy in the reaction coordinate. For a substrate to become transition state, irrespective of whether the reaction is endergonic or exergonic, you need energy. Okay. Conversion of substrate to transition state is always an endergonic reaction. All right. So now look at this. This is the free energy of substrate. This is the free energy of product. The difference between the free energy of product and substrate is delta G. Here the delta G is negative because this is an exergonic reaction. So this is your transition state, which is denoted by the symbol double dagger. The energy difference between the transition state and the substrate is delta G double dagger. Some people denotes this as EA, that is activation energy. So activation energy is the energy difference between the transition state and the substrate. So this is in context of a reaction happening without the help of enzyme. So when enzyme is involved, you can see that the transition state is obtained faster, right? So with the enzyme, this green color is showing with the enzyme. So with enzyme, the activation energy is decreased compared to without enzyme. So how is lowering of activation energy is possible? That is possible because enzyme binds to substrate, right? So there is formation of enzyme substrate complex. So during this binding energy is released. This energy is responsible for lowering of activation energy. Let me show you a graphical example. See this, enzyme binds to substrate through multiple weak non-covalent interactions. There is release of energy that is called binding energy. Because of that, substrate is converted to transition state. So you can see the transition state is always denoted by this symbol, that is double dagger. Here, let me tell you a very important point. The active site of enzyme is actually complementary to the transition state rather than the substrate. Okay, so transition state is stabilized, which finally leads to formation of product based on the thermodynamics. All right. I hope you have understood the concept of activation energy very clearly. If you have any doubts, feel free to ask me in the comment section. If you like this video, please share this with your friends. Thank you for watching. See you in next video.